So we are studying what pathway? Photosynthesis. Photosynthesis has two kind of endpoints. One that happens in the light. And what do we finish up in the light? What is the outcome? We made two things. When light shines upon the plant, it creates NADPH and ATP. There was a waste product of oxygen. Yes. And so the plant was producing the NADPH and the ATP. By chance, it gave off the oxygen. Important to us. All right. And so I have lots of energy molecules. Now, how was that energy extracted from the sun? Give me a big principle. How in the world did I take the sun's energy and make those two molecules? Photons of light excited electrons. Electrons flowed. There was kind of an electron transport chain, a chain of events where the electrons flowed, and all of those electrons flowed, ended up creating a proton gradient, so ATP could be made, and the end product of where the electrons flowed ended up into NADPH. Where did the electrons come from? That flow. Where do they start from? If we think about the entire chain of where electrons are moving from and to. We ended up being at NADPH, that's the end, but where did they start from? They started, it's in the chlorophyll where they are, but it comes from one organic structure. P680. P680 was the one that started the jump, right? But that wasn't actually the absolute beginning. It was the start of the movement. But once P680 left, I had to refund that electron, right? Where did the electron come from? It came from a manganese complex called the oxygen evolving complex. And those electrons came from the manganese center reacting with water. So we strip the electrons out of water and we allow them to go through this process. Now, how the electrons get moved is by the photons of light reacting with that special pair. Okay? Now, the chlorophyll absorbs lots and lots of energy, transfers it around until it gets to the special pairs and allows the separation of charge. All right. The second set of reactions are confusingly sometimes referred to as the dark reactions, although they can happen in the light. Okay? These are reactions that are not absolutely dependent upon the light, but they are dependent upon the products of what the light produces. Okay? And so the outcome that's going to happen is I'm going to make carbohydrates. Okay? I'm going to use the ATP. I'm going to use the NADPH, and I'm going to start off with a product or a reactant that's in the atmosphere, and that is carbon dioxide. Okay? And so we're going to do the synthesis. All right. The photosynthetic carbohydrate synthesis is named a whole bunch of things just to make it confusing. It's named oxygen fixation. We're going to fix oxygen into a larger molecule. Carbon dioxide assimilation. We're going to assimilate it. Or just carbon fixation. Or Calvin cycle. Now, why do we call it the Calvin cycle? Because Dr. Calvin was the head of the lab group that all of the graduate students did the work, and he took all the credit. Okay? That's kind of how it works in science. And so he was one that was credited with looking at and determining most of the reactions that occur in this. All right, in this particular photosynthetic pathway, there are three main stages in which reactions or separate reactions occur. Stage one is the actual fixation, where I actually connect carbohydrate, the carbohydrate to a CO2. All right. Then we will create in stage two. This is where we're going to use all the energy. We're going to reduce them, okay? Because CO2 is very oxidized, the sugar is reduced, and so we have to invest power. This is where we invest all that ATP and all that NADPH that we produced in this particular step, and then we create a molecule right here. Tell me what pathway this molecule represents. Oh, we need to make sure we know where we're pathways. Okay, you're going to say glycolysis, but what? But it's glycolysis, we know, is glycolysis synthesis? No, glycolysis isn't synthesis. This is represented in the glycolysis, but what's the reverse pathway of glycolysis? Gluconeogenesis, the synthetic pathway to synthesizing glucose. All right, so we will end up with some of this. Now, think for me, if by chance you were right here, how many glucoses could you make? Lean to your partner. Talk about it for a second. How many glucoses could you make with that starting point? Okay, do you got a guess? We're going to take a theoretical guess. Okay, we're going to make a hypothesis. How many glucoses could you possibly make? Okay, anybody say 1? Anybody say 2? Anybody say 3? Okay, I've got 1, 2, 3, 4, 5. Anybody say 4? 5? 6? Could you make 6 glucoses from there? How many carbons are in 6 glucoses? How many carbons are in 1 glucose? Let's start off so. How many? 6. If I had 6 glucoses, I would have how many carbons? 36. How many carbons do I have right here? I only have how many? 3, but 6, 18. Okay. And so if I have 18 carbons, the most I could have ever made was 3 glucoses. Okay? If you took those, it was too simple, I know. If you took those, all of those right there, and you made 3 glucoses, okay, the plant would die. Okay? Because you have taken in, at this point, 3 CO2s. You've gone through this. You have 18 carbons that are potential. This is an intermediate of gluconeogenesis. I could make sugar from all of them. And if I took all 6 of them and moved them down that pathway, I'd make 3 glucoses and die, because I wouldn't be able to fix anything else. I wouldn't be able to incorporate any more CO2 into the plant. So this is what has to happen. I can take one of them as an extra. Okay? I have to take 5 of them and rearrange them into the starting material. And so if I do some stoichiometry here, ribulose 1,5-bisphosphate is a 5-carbon intermediate, and I have 3 of them, so I have 15 carbons right there. I add 3 carbons right here. That means I have an intermediate that has 1, 2, 3 carbons, right? I have 6 of them. 3 times 6 is 18 carbons. And look, lo and behold, 15 plus 3 is 18. Great. All right, in the reduction, this represents still 18 carbons. That's great. This still represents 18 carbons. That's great. This right here represents 3 carbons. Okay. We did see the transformation of glyceraldehyde 3-phosphate, okay? And we can manipulate that and make it dihydroxyacetone phosphate, and we can do synthesis. But up here, this represents 15 carbons. Those 15 carbons are not in the right positions and not in the right molecules to allow CO2 to be incorporated. So that leads us to the last stage, regeneration. I take those 15 carbons and I do some reactions to make it what I need to start the process. All right? And so those are our 3 separate processes. Now, stage 1 uses 1 enzyme. Okay? So one simple, one and done. Okay? The stage 2 reduction has a couple of enzymes to do. And I have to have an enzyme that uses ATP, and then I have to have an enzyme that uses NADPH, and I get that done. All right? And then I might have to isomerize a little bit. Stage 3 becomes a very complex set of reactions that will regenerate my starting materials. Okay? I will not go into as much detail in that, but I do want you to understand the stoichiometry between those. Okay. Stage 1 is the fixation step. It is completed by ribulose 1,5-bisphosphate carboxylase. Okay? Just because we're biochemists and scientists, we're lazy, so we call that Robusco. All right? And so, what does it say? It says, I'm going to take the molecule ribulose 1,5-bisphosphate. Okay? The bis, I have 2 phosphates in the 1 and 5th position. And I'm going to carboxylate it. I'm going to put a carboxy moiety on it. And we have seen this before. If a acid comes off, it becomes a CO2. If you add a CO2 onto something, it will become an acid. That is how it works. Now, this mechanism I don't like because they did it wrong. Okay? This step, there is a lysine that becomes important, but lysines actually are supposed to have a nitrogen that is charged, right? But what's being reacted here is not the lysine itself. It's a carbamylated lysine, and magnesium's a part of the reaction. We'll show this in just a moment later how that works. So the lysine has a modification. I have a negative charge. This acts as the base. I create a double bond, and I kick up onto a negative charge. Really, this negative charge, they should have said that it picks up a proton, because you see a proton there, the hydroxide in the next step. What they didn't show was a base. Some amino acid inside the side chain will take the proton. I will kick the electrons down, and I would have liked to show that that pair of electrons was the one that attacked. Okay, there's the second step, the carboxylation. I have had a CO2 floating around as a gas, and now it is an acid. Again, to facilitate the reaction, I make the water a better nucleophile by deprotonating it, and I would rather show that pair of electrons as attacking. All right. It does show, the artist in this particular case does show the negative charge, which is the transition state. It will come back down, and this will be a leaving group, and that is wonderful. In the final process, this carbanion takes up a proton. Now, most likely what's going to happen is that this proton from that lysine is really, really close. And so when this kicks down, most likely this picks it up very, very quickly, so that it doesn't float around as the carbanion, which is very high in energy, but they did articulate it for you to show that it exists. All right. This is the addition of 1 CO2. In the stoichiometry that we looked in that circle, we would have to do this 3 times. We produce in each one 1, 2, 3 phosphoglycerates. If I go from the stoichiometry from the circle, I will have produced 6. All right. And so if we go from the stoichiometry from the big circle, we start off with 6 of these, and we'll end up with 6 of these. And so we look at an enzyme called phosphoglycerate kinase. All right. Talk to me about what you see in that reaction and what you might think about the mechanism of this. It is a kinase, yes. So that kind of gives you an idea what class the enzyme is. All right. Where would the nucleophilic attack happen on the ATP? On the gamma phosphate. The reason you can say that is you see one phosphate that is transferred, and the indicative part, ADP, is the one that leaves. And that tells you that's why it has to attack at the gamma phosphate. And so I would attack at the gamma phosphate and have that transferred. Why in the world would I put a phosphate on there? Why would I use that energy? Why do we normally put phosphates on? To make a what? To make a better leaving group. That is fundamental in our synthetic processes. We also saw phosphates put on proteins to control activity. This is a reaction, not a protein, so we're not controlling activity, but we are activating a wonderful leaving group. Now, that means I have to have something that wants to attack and a place that wants to be attacked. This right here wants to be attacked. This is an electrophile because it is partially positive. Why is that carbon partially positive? Because of the oxygen, the electronegativity difference. What is the nucleophile? It is the hydride that comes off, something that is negative. And when we see NADPH, the normal reaction for an NADPH is for a hydride to leave and to become a nucleophile. And the phosphate falls off. Now, to do that, I invested energy in the kinase, and I invested energy in this glyceraldehyde 3-phosphate dehydrogenase. The dehydrogenase part of that name tells you hydrides most likely are going to be moving. And I get glyceraldehyde 3-phosphate, okay, which is what we saw as the product, okay, and I said we can end up with 6. We can end up with 6 of these, or we can end up with 6 of those, dihydroxyacetone phosphate. Have you seen this particular reaction before? Going from glyceraldehyde 3-phosphate to dihydroxyacetone phosphate? Yes, in what pathway? Glycolysis is half right. The other one would be gluconeogenesis. Okay, and so those can be in equilibrium, and they are in equilibrium. Okay. Don't worry. That looks a little bit complex. We're going to start talking about some of the enzymes in the next chapter. So don't worry, we'll learn these reactions, okay, but not for this part. What I'm going to do is identify, there's an enzyme called transaldolase that gets used. Here's transaldolase again. Here's, where's another one? I think that's it. Okay, and we're going to learn an enzyme called transketolase, in which I'm going to transfer a certain number of carbons. Transaldolase will transfer 3 carbons around. And so if you see this up here, 3 and 3 become 6. I transfer 3 of them onto an entity that's going to accept. If I look at this transaldolase, I have 1, 2, 3, 4, 1, 2, 3. I will transfer these 3 onto the 4 to make a 7. Okay, so transaldylases are a transfer of 3. Transketolases will transfer 2 carbons. And so I have here 6, and I have 3. If I take 2 of them away from here, put them on there, I get a 5. And if I take 2 away, I get a 4. And what I really need to end up with is 5 carbon intermediates. Okay, we need to get back to the 5 ribulose 1,5-bisphosphates. All right, so we'll learn those particular enzymes. All right, what does this kind of enzyme do right there? Not that particular one, but that class. We saw one in our last chapter. So the class of enzyme there is called an epimerase, an epimer. Do you remember where an epimerase was used? Yeah, it's like up or down on what? Up or down. That's a good. On a sugar, there is, if you have 2 sugars and 1 hydroxide, one is sticking above the ring and one is sticking below the ring, that kind of relationship is called an epimer. There are 2 epimers that you know of or should know of. One is galactose and one is glucose. We used an epimerase to change galactose into glucose. Okay, so that's all I'm going to do is change 1 rotation around a hydroxide. Okay, so that class is used. And it looks like I used that a couple times. Need to do some epimerasing. Okay. And then if you look at this enzyme by its name, the class, what's going to happen with that enzyme? What kind of class is that? Okay, it is in the big class called phosphatase. Now, phosphatase takes off a phosphate. It's the opposite of a kinase. Kinase puts it on. And I can look at this enzyme here. That's a kinase. It's going to put on a phosphate. So, at this moment in time, I'm going to not require you to know how this transformation happens, okay, but that it does happen. That we start off with 1, 2, 3, 4, and 5, 3 carbon intermediates. Okay. And we make 1, 2. Oh, wait a minute. Yeah, 1, 2, and 3, because there is. Let me do this. Let me erase this one. Do you see? There's 2 arrows. There's 1 at the top. So 1, 2, and 3 ribulose 1,5-bisphosphates. So I start off with 15 carbons at the beginning, and I end up with 15 carbons at the end, in the right conformation to start that circle. I might ask you, based upon the class of enzyme, what it might do. So in this case, you should understand what a kinase does, what a phosphatase does, what a epimerase does, and the transaldolase and the transketolase I'm going to teach you in more detail in the next chapter in another pathway. Okay? So that's as much detail as I want from you for this at this moment in time. Okay. And so what happens is that light comes in, photosynthesis happens, I create lots and lots of ATPs, okay? And I go through the Calvin cycles, and to make 1 3-carbon intermediate, I have invested, it says, 9 ATPs. Now let's go back and take a look. Really? Did I use 9? Okay. I used 6 here, and I used 3 there to produce, ultimately, this one extra 3-carbon intermediate that came from 3 CO2s that came in. So I used 9 of them. That's a whole lot of investment. Not only did I use 9 of them, but I also used 6 NADPHs, which represents energy too. Okay, and that's great. All right. Now the light in here is happening inside the stroma. The light gets absorbed. All of this synthesis is happening. And in the photosynthesis, if there's ADPs and PIs, I can make lots of these as long as light's happening, right? But to make sucrose, okay, the nice sweet sugar, the dihydroxyacetone has to be transported out of the stroma, okay, and it pulls it this way. If by chance I just had this happening and I made lots and lots of sugars and I only had this bottom part happening, photosynthesis would stop, because with every one of these that is transported out, I take away a phosphate. Okay? And phosphate is made in this compartment. ATP synthesis is not in this compartment. And so the balance is, for every potential sugar molecule that goes out, a PI has to come back in so that you can continue to make ATP. One of the limiting factors for how well a plant grows is the amount of phosphate available. Has anybody gardened before? Okay, so have you ever bought fertilizer? And one of the key components in fertilizer is a concentration of what? Phosphate. That's one of the rate limiting steps in terms of how well a plant can actually grow. You need a lot of phosphate because, quite frankly, there is so much sun energy in the daytime that it peaks the capacity of all of the intermediates of the step, and it's always running. And you just limit, you could actually have created more energy molecules if you just had more phosphates and ADPs, and you could do more synthesis. And so sometimes the lack of phosphate in the soil can limit growth of plants. Okay. Now, how do we regulate this? All right. The Robusco, how in the world do we get this done? All right. Robusco actually binds to ribulose 1,5-bisphosphate, and that's a substrate, right? I use ribulose 1,5-bisphosphate and then add a CO2. That's what's going to happen. Now, this is the oddball thing. In the dark, if the ribulose 1,5-bisphosphate binds to Robusco, it will bind to it and inhibit it. The substrate inhibits the enzyme, and it sits there and it blocks the active site. When the sun comes up, okay, there is another enzyme called Robusco activase, which is kind of a direct, you know what that thing's going to do. It uses ATP and it removes the ribulose 1,5-bisphosphate. And what happens is the lysine that we saw that was the first reacted part is now able to react, and the activase not only moves the ribulose 1,5-bisphosphate out of the way, but it also allows CO2 and magnesium to come in and the CO2 to react with the lysine. It only takes the enzyme once to do it, and then the enzyme can complete the entire sequence and react and react and react and react until the light goes away. The CO2 that is attached to that lysine, all right, has to be positioned to take the proton that we want off. Well, how does that do that? Well, that magnesium that comes in actually coordinates to the oxygens and helps bind the substrate in the right conformation. Now, we've talked about this, but we're showing you here particularly that an enzyme will bind the substrate in the right three-dimensional structure. Now, only in the case if there's magnesium and CO2 that's reacted with it, will it bind it in a way that it is reactive. If the CO2 that reacted with the lysine and the magnesium is not there, Robusco would bind to it in a whole different conformation and block its activity. This is the first time we've seen a substrate act as an inhibitor. Okay? And so we can see the beginnings of that reaction is oriented by the magnesium, a cofactor, and the CO2, which is a cofactor, that changes the lysine's charge from a positive charge that is interacting with the species to a negative charge, making it act as a base. All right. So what happens in the differences between dark and light? All right. So in the dark, after the cell has used up all of the ATP and all the NADPH that it has that excess to, and those concentrations become really, really low, that tends to make every synthetic place stop, okay? And it kind of gets reset. What happens is, without the sun, the pH in the chloroplast or in the stroma and in the lumen become equal, because the light is what builds the gradient. Okay, I let it go through, make ATP, and if it's not building the gradient, always going down, it'll reach an equilibrium. All of these enzymes that are part of the Calvin cycle stop. Okay, when light shines, what happens as the photons are happening, you get changes in the stroma. Okay, concentrations change. First off, the flow of electrons through the process make the pH much lower or more basic than inside. So pH change happens. That's one of the signaling. Okay. This pumping. Also, NADPH concentrations go up. That's a signal to make things go. Additionally, ATP is produced. That's a signal. What we're doing is we're going to compare the concentrations of PI that are in the dark and the concentration of PI and ATP in the light, and that will cause things to happen. One other thing, in the light process, magnesiums are pumped out into the area where synthesis happens. We just looked at Robusco. Robusco needed that magnesium, so the concentrations of magnesium go up high enough so that it can be part of that particular reaction. In this slide here, there is one more entity. You get ferredoxin in high concentrations. So we get ferredoxin here in high concentrations in the stroma. Okay? And it's in the reduced form. All right? Each one of these players are going to turn on or signal the enzymes for making sugar to go. Okay. The ferredoxin, okay, if we go back and remember what normally happens to ferredoxin. Normally, ferredoxin gives its electrons to the reductase. Okay? And I make NADP. Okay, now I've made B. NADPH. Okay, normally that's what I make. But you can overwhelm the NADPH. So what can happen is ferredoxin can get up in so high a concentration that it will go over and it will give its electrons to a reductase, a different one, which gives its electrons to thioredoxin, which then gives its electrons to the ribulose 5-phosphate kinase, the fructose 1,6-bisphosphatase, the acetohepulose bisphosphatase and the glyceraldehyde 3-phosphate dehydrogenase. These are part of the regeneration steps. This is part of the fixation. So these are stage 3 and stage 2 enzymes, and they only become active when they are reduced. They are only reduced if ferredoxin is in high concentration. Ferredoxin is only in high concentration when there is light. And so ferredoxin can go make NADPH. Ferredoxin can also go back to the cytochrome BF complex and allow a little cycle to run to make just ATP. Ferredoxin can also go signal the enzymes that say make carbohydrates. It makes them go. So ferredoxin's a really important player, and we will learn one more thing that ferredoxin does next semester. So ferredoxin is important in a lot of different places. All right. The Calvin cycle and the Robusco. It's a really elegant enzyme, but when we teach enzymes, we say enzymes will bind specifically to one substrate, right? They're kind of like, I only bind to this and I only do this reaction. That's kind of a hallmark of biological systems. That allows us to go to 100% completion in reactions where in organic chemistry, we boil the mess out of it and get lots of side products. All right? Robusco is elegant, but it's, you could term it a lot of things. Loose, non-discriminatory. It just likes to get together with more than one person, okay? And so just kind of a little out there. And so what you really want to have happen is, hopefully you'll remember it when I say it that way. What it really wants to do is ribulose 1,5-bisphosphate and react CO2. That's the normal reaction. But it can also have as a substrate the ribulose 1,5-bisphosphate and oxygen. It sometimes doesn't discriminate well between CO2 and oxygen. And so if it does pick up oxygen, it will create my 3-phosphoglycerate, which is I want, and I get a 2-phosphoglycerate. That is not what I want. That is kind of a waste product. I have to convert that, put it in the peroxisome, convert it, make it an amino acid, send it to the mitochondria, react it back to get an amino acid, send that amino acid back to the peroxisome, do a reaction, reconvert it into glycerate, go all the way back up to glycerate, use an extra ATP to make the second 3-phosphoglycerate. So I can go through this. This is a wasteful product. It's called photorespiration. It is taking the waste product and bringing in it. It's taking in oxygen, which is not what it really wants to do. All right. What increases this? Okay. It increases this kind of reaction if the conditions are warm and if oxygen concentrations are high. Okay. So where in the world is it warm? All the time. At the equator. You can say Cleveland, Tennessee, right now, okay, in the tropic zones and in the equatorial zones. And so in the tropical zones and equatorial zones, the plants would actually do a lot of phosphorespiration actually to the detriment of plants. And so those that are in the northern or southern latitudes usually have a certain particular structure that allows the reactions I just showed you called the C3 reactions. Okay? In the tropics, this is the reaction. It's the same cycle, but CO2 comes in, and you show the 3 and the 2-phosphoglycerate. In the tropics, plants have a different structure. Okay? Because the plants not only, if you think about the tropics and the shape of the earth, what is the shape of the earth? It's a what? It's kind of a circle, but it's three-dimensional circle, which is a sphere. Okay, good. The earth is not flat. All right. Now, is it a perfect sphere? No, it's kind of, because of the rotation, it's kind of squished. down, the tops and the bottoms. That means the equator is thicker, and our atmosphere is actually thicker in the equator. There's actually more oxygen at the equator than there is as you go north and south because of the shape and the rotation, the spinning of the Earth. And so that just kind of, it's warmer and it's higher in oxygen, and so it would be really bad. So what happened is that there are what are called C4 plants. It's a different physical manifestation of the plants, and what happens is this. Oxygen and CO2 can get into the cells of these, and what we do is we do a separate reaction with phosphoenol pyruvate, and we create oxaloacetate. We've seen this reaction in regenerating the citric acid cycle intermediate. The oxaloacetate, by happenstance, is a 4-carbon to-intermediate. This is why they are called the C4 plants. That is placed into the chloroplast. That gets converted into malate. Malate comes into another compartment, and I regenerate pyruvate, but at that moment in time, I give off CO2. The CO2 now, down here, is given off as a gas, and the concentration of CO2 is much higher than oxygen. Oxygen is pretty high up here, but I'm not doing the reactions necessary to create sugars. Okay, the pyruvate gets shuttled back up and creates the phosphoenol pyruvate so that this cycle can occur. I create the gas CO2 that can go into the Calvin cycle, and even though it's warm and has high oxygen content, I almost always go through the right reaction because the plant's physiology is different. And so we have some plants that are C4, some plants that are C3. Most of the plants that you see have the structures of C3 plants. All right. Now, we've talked at gluconeogenesis. We'll look at that, unfortunately, in the next chapter. So suffice it to say, glycolysis runs not in reverse, but parallel reactions that go reverse. And I'm going to make fructoses if I don't go all the way up, and I can make glucose if I go all the way up that synthetic pathway. Okay? Now, that's all well and good, but single sugars are never the end products of most plants. Okay? Most plants will take those single sugars and make at least disaccharides as a storage entity, but most of the time it's going to make long polymers for storage. Okay? And the polymers that are made inside plants are called starch. Do you know what the polymers of sugars inside the human body is called? How we store sugars. I haven't introduced it in this class, but you may have heard it in another class. It's a way we have a polymerized sugar store found in muscle and found in liver. It is called glycogen. They're very related, and we'll see that there's just a slight difference between the two. Okay? And so, if I get all these single sugars, I will probably make the starch as a storage molecule. Okay? Now, if I go back up glycolysis, will I ever make this molecule right here? Does glycolysis or gluconeogenesis have that molecule? No. What's the closest thing that we can get in glycolysis or gluconeogenesis? Glucose 6-phosphate. Okay? Glucose 6-phosphate is made, and then there is a mutase that converts glucose 6 into glucose 1-phosphate. Okay? So glucose 6-phosphate. My G and my 6 look very, very similar. Okay? Can be converted using a mutase into glucose 1-phosphate. That is the entity that starts down the pathway to make polymers of sugars. Okay? Now, I'm going to use energy to make this go. And if I'm going to make starch, okay, I'm going to use the power molecule ATP. That's all well and good, okay, because when it's sunny, I make lots of ATP, and so I have that to use. All right. My reaction between the glucose 1-phosphate and the ATP will produce ADP glucose. All right. Talk to your neighbor. How would the reaction occur to create that ADP glucose from the two reactants that you have, ATP and glucose 1-phosphate? In other words, where on the ATP is the attack and what attacks it? Okay. You have a 33% chance of getting this right, okay, at the outset. It's kind of like a test question, right? If you have a 33% chance of getting it right, that means you're going to have a real, what chance are you going to actually get it right? A 100% chance. That's exactly what I want you to think. Okay, so once you've learned everything, and you know, your first guess would probably be 100% right. It's when you talk yourself out of that right guess. Okay, where would I attack and what attacks? Okay. All right. Gamma. Who votes gamma? I got one hesitant half vote. Who says beta? I got 3 hesitant. 4. Who says alpha? I have a lot more. Okay. I haven't introduced this, but there's another option. I can actually attack the carbon, but we haven't even got there yet. But watch this. This phosphate will attack here. I'll kick up, I will kick down, and kick out. What does that represent right here? We write that as PPI. And so this sugar was the original on the 1. That phosphate. And this phosphate was the original on the ATP. Okay? We call it ADP, but we had 1 phosphate coming from the ATP and 1 phosphate coming from the glucose 1. All right? That was a little bit of a stretch to think through. I admit at this moment in time, but I want you to think about that. All right. Why in the world did I add that thing on? Why in the world do we use ATP most of the time in synthetic processes? To create a what? A good leaving group. What's the good leaving group going to be? ADP. And the ADP will fall off, and I will make a connection between this hydroxide here and this carbon there to make this 1,4 connection, and it will be in the alpha conformation, the alpha 1,4. And I do this again and again and again and over and over and over again, and make long strands of starches. What's interesting about this particular enzyme is we don't know the mechanism. We don't have a clear picture of it, but we do have this interesting idea, and we see this in a lot of biological synthetic pathways. The enzyme actually has 2 active sites in which the ADP with a glucose and an ADP with a glucose comes in. That's the activated species. Something will attack it, ADP leaves. Something will attack it, ADP leaves. So I charge it, or I put one sugar and one sugar. That isn't a polymer. Okay, that's two individual sugars connected to something. When the third one comes in, okay, what happens is I attack one half and take one of the other sugars. That allows this to take the third sugar and put it on waiting. So now I have a 2 polymer. This third sugar then attaches, and this then picks up the fourth. And what I do is I grow the chain back and forth between the two entities as it gets longer and longer and longer. We're going to see this with sugars. We're also going to see this with fat, that the interplay of the active sites work back and forth as they elongate. But we don't have a great crystal structure of this particular enzyme, and looking up the mechanism, we're not exactly sure how the mechanism occurs. That is one option for us to synthesize. The next option that we can make is sucrose. Sucrose will need to have a glucose and a fructose. Now, the fructose 6-phosphate comes from the glycolysis, gluconeogenesis pathway, okay? And we just don't go all the way up. How in the world do I get this glucose UDP? What is the power molecule here before I get to that? UTP. So I utilize a whole different pool of energy to go to sucrose rather than to go to starch. So our activated species is different. The energy is exactly the same, but having an A there or a U there binds it into a different enzyme for a different outcome. All right. We add this to be a good leaving group. Okay. A synthase is a molecule that connects carbons or entities together with the use of a triphosphate. And I will get the bond that I need between the 1 and the 2. But this is not the sugar that we eat because we have a phosphate on there. So we use an enzyme called a phosphatase to take that off and to make sucrose. When I lived in Puerto Rico, right outside the base was a sugarcane plant. You could stop by and just get a chopped off stalk of it. The sweetest thing you could ever imagine. The problem was it stank to high heaven every time you drove by it. It was just this really kind of oddball. It was sweet and smelled so bad because some of the product would rot out in the sun, and you'd get some decay as part of the process, as part of the natural process. That decay is important for the soil to be regenerated and to keep lively, but kind of interesting. That's a C4 plant, that the sugarcane, because it is in the tropics, and it can grow a lot of sucrose. How do I regulate whether I actually make sucrose or not? Okay. Well, there are things that will cause it to synthesize and not. Now, here are the two enzymes. Okay, sucrose 6-phosphate synthase. That is this one at the top. And then the phosphorylase, okay, that can react, and this is this actually should really be, I don't know why they did this. It should be U, because we have UDP glucose as the activated species. All right, think about things that would happen. In the light, we get lots and lots of ATP. Okay, so we also can make sugars, and so anything that is produced in the light, like glucose 6-phosphate, all right, tend to make the synthesis go. It also stopping the enzyme. Okay. In the dark, though, I don't have lots of ATP or UTP. I have lots of PI. I have lots of PI. PI tends to stop the conversion of this into an active state, and it will stop the actual enzyme itself, or it will stop the activating of glucose so that it can be reacted. Another energy molecule, if I have lots of 3-phosphoglycerate, that is made from the fixation. That is also indicating I have a lot of starting material. So if you have the things that are made in fixation or gluconeogenesis, it makes things go. And if you have the PI at the end, meaning that it's dark, I'm not making ATP, I'm breaking all my ATP down, it says stop synthesis. That seems to be logical in how it's organized.